that green beads, guanine, only pair with orange beads, cytosine, and red beads, adenine, only pair with pink beads, thymine. These are complementary base pairs. Notice how guanine and cytosine have a pink pipe cleaner. That represents 3 hydrogen bonds, while adenine and thymine have an orange pipe cleaner. That represents 2 hydrogen bonds. Now we will show you DNA replication. During DNA replication, an exact copy of each DNA molecule is created. Enzymes important to this process are DNA polymerase. Included in DNA polymerase is helicase, which separates the DNA double helix into 2 single strands by breaking hydrogen bonds between the complementary base pairs like this. Now free-floating nucleotides in the nucleoplasm form complementary base pairs with the unzipped DNA nucleotides and covalent bonds with each other. This is catalyzed by DNA polymerase. As you can see, the DNA polymerase work in opposite directions. As you can see, one strand of the replicated DNA molecule is from the original DNA molecule. This is the parent strand. The other strand formed with free-floating nucleotides is the new strand. DNA replication is a semi-conservative process because half of a pre-existing DNA molecule is always saved. Transcription is when part of a DNA molecule is unzipped with the help of RNA polymerase, which are enzymes. However, as mRNA is a single strand, only one strand of DNA is replicated. The strand that is replicated carries the genetic code and is called the sense strand. The other strand is the antisense strand, and this is used as the template for the mRNA. RNA polymerase helps free-floating RNA nucleotides float into place by complementary base pairing. On the mRNA, 3 bases are called a codon. Notice how the RNA has black beads because it has ribose sugar, and white beads because it has uracil instead of thymine. The mRNA created in transcription leaves through the nuclear pores and eventually bonds to a ribosome in the cytoplasm. This is a tRNA. On one end, it has 3 unpaired bases, called an anticodon. On the other, it has an amino acid attached. If the tRNA has an anticodon that is complementary to the mRNA's codon, it will bind to the site where the mRNA is. A second tRNA comes and matches its anticodon to the mRNA's codon. Then an enzyme catalyzes a condensation reaction between the 2 amino acids and a peptide bond forms. Then the ribosome moves and the tRNA molecule in the first site is pushed out and the new tRNA molecule enters the second site and bonds with the new codon. The new tRNA molecule brings in another amino acid and a peptide bond forms. Eventually, this process continues until a polypeptide is formed.